All right, Hey Bio 2. I'm giving the kind of supplemental lecture here. And what I kind of want to do with this one is kind of just walk through really briefly. We'll go into more details in class itself, but I just want to throw these different phylogenetic clades of different animals your way. So there's a lot of overlap, especially in the next week or two, between what we'll discuss in lecture and if you're taking the lab, what we're going over in the lab as well. And so I kind of want to highlight that by giving, especially to those who aren't taking the lab, giving you that relevant information here. So if you are taking the lab and have looked through the voice threads and the slides in lab, a lot of this might be familiar to you, but by me talking through these slides, maybe you'll get a little bit more information as well. So it might be worth listening for that reason alone. So because of that, I'm kind of showing you the animal diversity lab slides here. So animals fall under the category of eukaryotic organisms, just like plants and fungi. All animals are eukaryotic. They're multicellular heterotrophs, as opposed to plants, which we know are autotrophic. Animals are all heterotrophic and multicellular. They don't have any cell walls. So up until this point, we've talked about what different cell walls are made out of, like chitin in the form of fungi, cellulose in the form of plants. No cell walls here in animal cells. We'll see that there are a lot of different traits that animals have that kind of make them unique, but that most animals share. And importantly, they almost all have an ability to reproduce sexually. That's not to say that they all do reproduce sexually. We'll see a couple examples of asexual reproduction in some animal systems. And then we'll see, like in this animal group, we start to see the evolution of sensory structures and a nervous system. Again, we see this within animals, but it's not to say that all animals have these structures, as you'll see. In terms of animal development, we start with a zygote, a term that you saw with plants as well. Zygotes form starting off as a single cell, and it starts kind of multiplying from there, getting larger and larger through this process called cleavage, where cell division occurs at first without any growth. So essentially, you're seeing the same volume in here in figure A, where one cell becomes two cells half their size, then four cells a quarter the size of the original, etc., until finally it does start to grow after this phase of cleavage, in which case what's formed is called a blastula. And this term of blastula, the blastocele, you'll see these four letters COEL appear frequently. And you'll see this prefix blast appear frequently, and it's one way that animals divide themselves according to different clades. But in terms of the blastula itself, it's basically just this hollow ball of cells with fluid within the cavity. Now, once it's formed this blastula, what happens next is a process called gastrulation, which creates this sort of dent in the ball where it starts to fold in on itself, creating what's called a pore, specifically a blastopore. And so within the characteristic of this blastopore and the fate of the blastopore is one way we divide animals into protostomes and deuterostomes. So if this hole or pore that forms within the blastula is going to become one of two things in all animals. It will either become that animal's mouth or that animal's anus. And if it becomes the mouth, then we call that a protostome. And if it becomes an anus, we call that a deuterostome. And one way you could remember this is, you kind of pop a deuce from your anus. And so think of anus and deuce. That might just be one way you can remember which one is which. The deuterostome becomes the anus. You might have your own way of memorizing that though. Now, sometimes we can categorize animals according to a body plan, the way it essentially develops. And the three kind of ways we can do this that we see animals broken up to within phylogenies is by the types of tissues they have, first of all, the type of symmetry they'll have, we'll go over that in a second, and the type of body cavities that are formed, this protostome versus deuterostome distinction being an example of that. Now, regarding the tissues, we don't see this within the these terms up here when I go through the phylogeny, but we'll see that it basically coincides with different types of animals. So there's diploblastic tissues, and all this means is that there are two germ layers formed. Essentially, the exterior and then the wall that kind of lines the animal's gut. So these two germ layers, there are only two germ layers in diploblastic animals. And we see this most typically in animals with radial symmetry. I'll go over that in a second. And then there's triploblastic, which is essentially just three germ layers. There's all the cells that line a gut. There's also all the cells that line the exterior, like the skin or the cuticle, depending on the animal you're talking about. And then there's an internal layer called the mesoderm. And this is most commonly seen in animals with bilateral symmetry. And this mesoderm, as you see in kind of point 3 here, is what becomes an animal's skeleton, muscles, kidneys, all that. Essentially what both diploblastic and triploblastic animals both have are these what are called the endoderm and ectoderm germ layers. So the ectoderm is just what ends up becoming all your external features, like your skin, also your jaws and teeth, and the nervous system in animals that have it. And then the endoderm is just all your digestive tract lining, as well as a couple important respiratory system features like lungs. Now, the second way we can divide animals is through symmetry, and we'll see this in the phylogeny pretty clearly. There are animals that, first of all, have no symmetry whatsoever. We'll see that in the most basal form of animals, the porifera, what you might know as the sponges. Then we have animals with radial symmetry, and basically what it means is you can take a cross section from kind of the top down and have these two symmetrical parts, no matter which way you cut it. Think of things like a jellyfish. If you were to look at a jellyfish from the top, any way you cut it, it's going to look symmetrical. The same goes for most anemone. And then we'll see kind of an exception to this rule, which are starfish. They aren't within the same family of animals as radial symmetry, and I'll explain why that is a little bit later. And then there are all the rest of the animals, which show bilateral symmetry. Now, as opposed with radial symmetry, you can kind of cut it in half, cut the animal in half either way, and you're always going to have two symmetrical sides. With bilateral symmetry, there's only a couple ways you could cut the animal to kind of create two similar halves. So when we're kind of orienting ourselves with animals, with bilateral animals, we refer to their tops as the dorsal side and the bottoms as the ventral. And then you can also further distinguish them in terms of anterior portions where the mouth and eye parts will often be, and the posterior end, which will be where the tail and anus are. And then this last way we can divvy up animals is through their body cavity. So we already saw that you can become a protostome or a deuterostome depending on which body part the blastopore forms. But the cavity within the body is referred to as the coelom. We saw these four letters, COEL, first mentioned when the blastula is formed. That internal cavity is the blastocele, which becomes the coelom. And the reason we have body cavities is essentially for our own protection, cushioning the organs we have, as well as for moving water around in our system. And so there are three types of body cavities that animals can have. It's basically, the easiest one are acoelomates, which have no body cavity whatsoever. All they have is, their digestive tract, that's hollow inside because food kind of has to pass through it. So there has to be some cavity, but there's no body cavity other than that, really. Everything else is kind of tissue. Endoderm, which is the line of cells lining the digestive tract. Then the mesoderm is kind of everything between the digestive tract and the skin, the ectoderm. So one animal example of this are flatworms, with eucoelomates, which are the next ones. These are eucoelomate, meaning true coelom, true body cavity. I don't really like this diagram because here it makes it look like it's all solid, but what's shown in green here is kind of hollow body cavity space. This is where the coelom is formed. And so here you basically have, your digestive tract along with the mesoderm, but within the mesoderm, there are these body cavity spaces. And so you can think of it in terms of us. We are eucoelomates in this sense. We have body cavities, space between our organs and our skin. In the Bio2 lab this week and next week, pretty much all the animals we're dealing with are eucoelomates. So the earthworm that you might dissect, the clam, the crayfish, those are all eucoelomates. And then the last one is pseudocoelomate. Pseudocoelomate, what it essentially is that the digestive lining isn't held up by anything. There's a full cavity wrapping all around the endoderm, that digestive tract, as opposed to having some connective tissue within that holds things into place, which is kind of what our muscles do. So animal phylogenies have obviously changed over time. And when we look at the phylogeny, today we'll kind of go in terms of most ancestral or most basal of organisms, and work our way up in terms of general complexities that we see in animals. So the first things we'll see are really basic, the most basic definition of animal. And what exemplifies this are the sponges, which are the poriferids. Now, recent information kind of identifies this group, ctenophores, which are kind of like jellyfish, but not exactly. They're called comb jellies. They don't have the tentacles. They're kind of just, if you see them in Galveston, you can see them occasionally. They just look kind of snotty with no shape really to them, but they're technically the most basal form of animal, but will kind of go by the traditional sponge classification of this basal clade. So what all animals essentially are metazoans. From there, we also have eumetazoans. Now, eumetazoans are the true tissued samples, or true tissued animals, sorry. And they are basically just everything except for porifera, the sponges. Most of these animals you'll see are within the bilateria clade, which means they almost all have bilateral symmetry. The only exception is one group known as the radiata, and we'll get to them. This includes things like anemones and jellyfish. And then one thing that may surprise you is that most animals are actually invertebrates. So vertebrates only make up this tiny group within chordata, and not even all chordata, as you'll see. So when you think of animals, you often think of bears or dogs, us, but the truth is this is really just a tiny section of all animals within the kingdom Animalia. So remember, a clade is, we're going to see this term a lot. So remember that a clade is an ancestor and all of its descendants. So if you're looking at a phylogeny, it's basically one point of the branch and then everything that branches off from it and everything downstream of it. So Porifera and Parazoa refers to the clade of animals that don't have true tissues, the Parazoa. These are the only animals without tissues, and they consist primarily of the sponges. The way the life cycle works is the larvae of sponges are actually free swimming, but once they become adults, they become this sessile, we say, or immobile, you might also hear of it, structure that kind of just forms. And, if you didn't know, you wouldn't guess it was an animal, but sponges are indeed the most basal form of animals. So how does a sponge even work? It's often hard to think of these as animals, but again, they are. And what they have are essentially these pores where water goes in and gets filtered. It gets filtered through these kyanocyte cells. And what these cells do is they kind of filter the water and pass any incoming nutrients to the interior of the sponge, where there are these amoebocyte cells. And then within these amoebocyte cells, the nutrients are processed and broken down in this process called intracellular digestion, meaning digestion that occurs within the cell, intracellular. And then any leftover water just passes through and goes out through what's called the osculum, which if you see pictures of sponges, those holes that you can actually see are the oscula, the osculi, and then the pores are almost microscopic. So like I said, sponges and parazoans don't have tissues. So what do they have? So if you've ever touched a sponge, the actual animal, not the thing in your kitchen sink, you'd know that they're super rough, hard, and rigid structures. And the way they get this structure is through the formation of what are called spicules. So they don't have tissue. Instead, they have these kind of crystallized structures. If you look closely in this slide, they almost look like the Mercedes car logo. And these kind of are really hard compounds that, when clustered together, help give the sponge its rigid structure. So those are the only non-tissue animals, the sponges. From there, everything else has tissue. And within this clade of all tissued animals, Eumetazoa, we break it up into two of the next biggest clades, which are the Bilateria, the bilateral animals, and the Radiata, the animals with radial symmetry. So Radiata used to have this ctenophore group, but recent information has kind of moved it out of there. So now they're just the phylum Cnidaria. So this is pronounced Cnidaria. The C is silent. And these include your jellyfish, sea anemones, as well as some other animals like hydras, not the multi-headed creature from Marvel, although you'll see a picture and you can kind of see where the idea of a hydra comes from. So Cnidaria, essentially, it's become this wide group of things. Jellyfish and anemones, you don't necessarily think of them as the same thing, but they do fall under the same phylum. And it can kind of be summarized by its two different life stages. The first is the polyp. And the polyp is essentially just this sessile, that term again, just meaning immobile structure, where it's kind of stuck to something, some substrate, and then it has these tentacles sticking out. The tentacles surround what are both the mouth and the anus. For jellyfish and anemones, it's all the same hole. And then when it enters its mobile phase, it forms what's called the medusa. Basically, all that happens is once it's mobile, it kind of goes upside down. So you can imagine, a polyp upside down kind of looks like the tentacle, where you have the dome and all the tentacles kind of floating downward from there. This is just another picture of that, where basically you can kind of see the polyp stage of different, this Obelia species, and then it forms this microscopic jellyfish here. So many invertebrates, and I kind of highlight this here within the Cnidaria phylum, but other animals do this too. Many invertebrates can reproduce asexually through this process, Cnidaria do it particularly through this process called budding, where essentially a clone of itself just forms from the original, parent, you could call it. And then when it buds off of the organism, it becomes this free living clone of the parent. So the rest of eumetazoans are within the Bilateria clade, all the bilateral symmetric animals. From this clade, we can break it down into two more sections. So you'll see we're going to break things down into two more sections over and over again until we get to the whole of Animalia, or the whole of metazoans. So from this eumetazoa clade, or from this Bilateria clade, we can break it down into protostomes and deuterostomes, the protostomia and the deuterostomia. And the main thing that differentiates these two groups is, again, that blastopore that forms during the process of gastrulation. So I'm repeating these vocab terms so that some of them kind of stick. Gastrulation is the process of forming that cavity of the blastula. So remember, if the blastopore becomes a mouth, you've got a protostome. If it becomes an anus and can start chunking deuces, it becomes the deuterostome. So within the protostomes, the ones whose blastopore becomes a mouth, these protostomes, we can divide this yet again into the ecdysozoa and the Lophotrochozoa. Weird names, but you'll see that you've been introduced to both of these in the lab, or any day you walk around. So again, they both might sound incredibly strange, but many of y'all are familiar with at least some members within these two groups. So first of all, the Lophotrochozoa. We have a couple organisms we looked at in lab this week. The earthworms, which are here in the annelid grouping. And then we also have the phylum Mollusca, where we have our clams, snails, other things like that, the mollusks. Oh, and the name Lophotrochozoa comes from this feeding apparatus that all larvae have, called the lophophore, or the larvae have a trochophore, and the feeding apparatus is this lophophore. It's basically this ciliated structure, so it's got these like tiny tentacles within the feeding apparatus of some of these organisms. So within these, there are a lot of examples of different, it's a widely diverse group, the Lophotrochozoans. We have things like Platyhelminthes. So Platyhelminthes, the coolest thing about them is that these are the animals where kind of we believe cephalization came from. So cephalic coming from the head. So this is essentially the first group of animals that evolved a head-like structure. Obviously, this doesn't look like my head, but what we mean by this is that essentially we kind of get this concentration of nerves, what ultimately becomes a brain. Those start to appear in this group. Planaria is an example of a platyhelminth. We also get ocelli, which are sensory cells that are photoreceptive, kind of the basal form of an eye. Some popular animals within this, some infamous ones are parasitic tapeworms. They attach to their vertebrate hosts through their scolex. Here you can kind of start to see that ciliated mouth structure when I was talking about the lophophore. Um, and tapeworms are parasites that we ourselves can get too, from eating undercooked meat and things like that. Now, maybe the more famous kind of member within this group are the phylum Mollusca. So the mollusks, things that become snails, clams, lovely little seafood creatures like that, as well as squids and octopus fall into this phylum as well, although it's maybe not as obvious since they aren't like bivalves or things like that. But the three main parts to kind of most mollusk bodies are the visceral mass that they have, the mantle, which is just in biolab, I kind of showed this to you. So the visceral mass is just where most of its vital organs are. The mantle is what helps create its hard shell. And then the muscular foot is the way it moves around. And so we see this throughout mollusca. They are mobile, even though some animals you might not think of as mobile, but things like clams, those can move, and obviously snails can as well. It's hard to think of them as having cephalization sometimes. It's obvious with snails, but as we saw in the Bio2 lab, we do see mouth-like structures in clams, and this is getting at that cephalization. The other Lophotrochozoan that we saw is the earthworm, which is within the phylum Annelida, annelids. So earthworms and leeches are kind of the most popular or common forms of this, but there are also sea creatures called feather dusters that you might have seen maybe on Planet Earth or some nature documentary. Rarely do you see them in person, but maybe you have. But earthworms and leeches, we all kind of know what those are. So we went through a lot of this in lab, but for those who aren't familiar, earthworms have many of the most basic structures that we ourselves have. Especially when it comes to the digestive tracts. They have organs that you see in more complex organisms like ourselves. They have a pharynx, they have a long esophagus, as well as an intestinal tract. They also have an actual brain. Granted, in earthworms, it's extremely tiny. You can just barely see it on this slide here. And you probably, if you took the lab, are now familiar with this image in your head from dissecting your own earthworms. If you haven't yet, you might do it next week. So those are the Lophotrochozoans. From there, we go to the ectozoans or ecdysozoa. These are the two main category or phylum within these are Nematoda, so nematodes and roundworms, and then the arthropods, insects, spiders, things like that. The name comes from this term of ecdysis. Ecdysis is just what we refer to as molting. So when insects molt and shed their exoskeleton and develop a new one, this process is called ecdysis. So within the phylum Nematoda, these are the roundworms and nematodes. They have a pseudocoelom, one of those types of body cavities that I mentioned. So not a eucoelom or a coelom where there is no body cavity. They have a body cavity referred to as a pseudocoelom. And many of these species are parasites. Maybe the most or one you're most familiar with, if you have a dog in particular, is heartworm. They are mobile. They're triploblastic, so they have both endoderms, mesoderms, and ectoderms. And just like everything kind of a little bit more basal from it and everything more complex, they have cephalization as well. We're talking about protostomes, they are protostomes, and they have what's called a cuticle, which is just this protective layer that helps them retain water and protect them from nasty pathogens. So the other member, oh, my light went out. So the other group are the arthropods. So the ones you might be most familiar with are insects, But outside of just insects, we also have crustaceans, chelicerates. If you don't know that spiders aren't insects, they're not. They fall under chelicerates, and that one might be obvious, spiders, but maybe less obvious are the fact that ticks and mites are not insects either. They are chelicerates. You also have your centipedes and millipedes within this group, all your crustaceans, so the rest of the yummy seafoods that aren't fish or mollusks, so crabs, lobsters, shrimp, etc. And then the hexapods themselves, which are the insects. So lots of cool things within arthropods that start to get, we start to see in this group, things like jointed appendages kind of standing out. And then we really start to see more, specialization of the head itself. So, they have cephalization, but in these arthropods, we really start to see it, develop into what we would recognize even as a head. And so that was all the protostomes. And then the last group, we can talk about are the deuterostomes. So remember, the ones where the blastopore develops into the anus first. And this includes echinoderms, which are starfish, sea urchins, sand dollars, things like that, as well as chordates. So all of the vertebrates just fall into this one small category. And we're the only other animals that are deuterostomes are starfish like us. So, it might be hard to make comparisons, but we're more closely related to things like starfish and sea cucumbers than we are to insects. So this is just reiterating that here. The echinodermata, the name echinoderm comes from echino meaning spiny and derm meaning referring to the skin. So it's these spiny or spiky skinned animals, things with really hard ectoderm cell layers. Again, they move, and in the General Bio2 lab, I kind of walk you through how starfish themselves move. They have an interesting way of doing that. The other thing to note is that even though they look like they should fall under the radiata category, those radial symmetric animals, they are actually within Bilateria. And the reason that is because it's actually the larvae of these animals start off as bilaterally symmetrical animals. And so what they have is what we call modified radial symmetry, where as they mature from larvae into adults, they develop a radial symmetry, but they don't start off with one. They start off with bilateral symmetry. And then the last one are the chordates, the ones that we might be most familiar with. Vertebrates are within chordata, but not all chordates are vertebrates, but all vertebrates are chordates. That might be important for you to know later. Vertebrates are within chordata. All vertebrates are chordates, not the other way around. And chordata have 4 key distinguishing kind of innovations that we only see in chordata, but it's also important to note that all chordates have these 4 characteristics, even though we don't think of ourselves as having something like pharyngeal slits or gills. We do at some point in our development, very early in our embryonic stages. So the first, kind of breaking each of these traits down, the first one is the notochord. It's this flexible rod that ultimately becomes, our vertebrate spine. So, not all chordates have as complex a structure as we do, but they all have this flexible rod-like structure between the digestive tube and the nerve cord. Kind of gives us a rigid support along most of the length of our bodies. Along those same lines, I mentioned the nerve cord, and that's another thing that develops in chordates, this dorsal hollow nerve cord to be specific. And the nerve cord in chordates develops into the central nervous system, the brain, the spinal cord, We see other animals that have a brain and spine, and a dorsal nerve that runs its whole length, but it's not this dorsal hollow nerve cord. So we see it in earthworms that they have, a brain and a long nerve cord, but it's specifically this dorsal hollow nerve cord that's unique to chordates. The third one are pharyngeal slits. This becomes the gills in many animals, but in other animals that don't have gills, it's what eventually become, like in humans, our ears, head, and neck. So, we have pharyngeal slits, almost gill-like structures early on in our development, but, early on in the womb, we lose those. But they help with gas exchange, oxygen, things like that. And then the last one is this idea of the postanal tail. Obviously, it's easy to think of in things like reptiles and horses, animals with tails. We don't have tails, but we have a tailbone, and that's a vestigial structure for humans. And even in some people have a small protrusion that they either keep or get surgically removed, and that's just a sign of our evolutionary past that we also have this postanal tail. For most animals, it helps in terms of balance as well as movement. For us, we didn't need any of that, so we lost it eventually. And so I'll leave that here. This is already long enough, but I hope some of these terms make a little bit more sense now, because you may see them on the prep quiz on Friday.